All right, when we left off, right before exams, we were talking about mitosis, which was the division of the nucleus and all the stuff inside it. And then we were also talking about cell division, right? In this place, cytokinesis. But remember that a cell spends most of its life in something called, oh my goodness, what a mess. That's not what it's called. Kidding. All right, so this is cytokinesis. This is mitosis. So most of a cell's life is actually in interphase. All right, and remember that interphase has three different parts. You have G1, you have S, and you have G2, right? And G1 is they're growing, they're kind of doing their thing. You know, they're growing larger, they're just getting ready for all of that growing. Okay, and everything that they are about to do. So remember we talked about these little checkpoints. So I am going to put little stop signs. This is a checkpoint where we have to check and make sure that everything is growing and properly. Okay? Then we have another checkpoint after G2, so we got a little stopping point here after G2 to make sure, remember this is where our DNA is going to replicate. So we got to make sure that we replicate it okay, everything's going to be fine, all is well. Then right after mitosis, but right before the cell completely breaks, we have our M checkpoint. Okay, and this is where we're going to check and make sure that we don't need to kill the cell. Okay, so at any point here, at any point in these stop signs or these checkpoints, we can stop mitosis at any point if things have gone bad. Okay, and what will happen is either the cell will just remain, like let's say in these two situations right here, the cell probably will just remain in interphase. So we have one option, which is if it goes bad, let's say if it doesn't pass the checkpoint. And I'll just draw our little pass, two S's. If it doesn't pass our checkpoint, then number one, it can just stay in interphase until it eventually dies. But if it is already divided, if it's in the M phase, then we have to go through apoptosis. And this is programmed cell death. So it's not accidental that the cell is dying. There's a reason, and it is designed in such a way in which it will die. Okay? Now, you guys have been reading a lot about Henrietta Lacks and her cancer, and we're going to continue to talk about it because there's so many different interesting facets about her cancer. But her type of cancer was cervical cancer. That's what it started off with. So we need to, first of all, know where the cervix is. So this is the female reproductive system. So here you have the ovaries. This is where we get our estrogen and where the egg is released from, fallopian tubes, and the uterus. The very, very bottom part of the uterus is called the cervix, and you can see its labeling right here, okay? And the cervix basically is what's going to open to let the baby out or to let menses out, okay? Now, if you have cervical cancer, what happens is you get tumors right here on the right here on the outside of your cervix, and they basically will penetrate all the way through into because the cervix is part of the uterus, it will penetrate all the way into the uterus And then, of course, you'll have bleeding, and you'll have a whole bunch of other stuff by the time it gets, when she was first diagnosed, she was probably very early stages. Here, where it says early stage IB, and over here, this is late stage. But by the end of her life, when we are looking at her, like in chapter 11, it started to bleed, and it has started to go outside of the cervix, infecting other areas, okay? So that is what cervical cancer is. But the question is, how did she get it? Okay, so how does one go about getting cancer? What exactly is cancer? So cancer at its very core is unregulated cell growth. That also can be unregulated cell division, growth and division. All right. So basically what cancer figures out a way to do is it figures out a way to fool all of these checkpoints, okay? So it's important for us to really understand the mechanism of these checkpoints. So how does a cell decide when to divide? So all cells sitting around, they're in this G1 phase, okay? So they are in a G1 phase, and they are going to get a cell signal called a proto-oncogene. And in your book, it said that these are positive cell regulators. Basically, this tells a cell to divide. All right? So at G1, it's saying, okay, you can go on and you can go ahead and divide. Okay? You can go and you can continue to divide. So at each one of these checkpoints, you have this proto-oncogene. This is a piece of DNA that creates a protein. Okay? And this protein is going to say, okay, you can go ahead and divide. Now, it is at each checkpoint, allowing it to go on to the next part of the cycle. All right? So if somehow, if a mistake somewhere causes this proto-oncogene to not turn on, then that means the cell cycle stops, okay? So the proto-oncogene, I'm just going to, like this, if it is on, then we'll say cell division. If it is off, then no cell division, okay? So a mutation in the DNA, for instance, over here could cause the proto-oncogene to turn off and stop the cell division. But what if, and this is how cancer gets us, what if this proto-oncogene is always on? If that proto-oncogene is always on, then this cell that's going through the cycle doesn't ever stop. No one stops to check it because it's going so fast through the cycle. And so you get cell after cell after cell after cell after cell, boom, 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 boom. And that, if you remember what that tumor looks like, is just a whole big old bunch of cells all in this, and it will create a tumor. Because you just have this cell that's this proto-oncogene that just is always on. It never turns off. Okay? So if it's always on, even a healthy cell that's not supposed to divide all of the time. So for instance, your liver cells don't divide all that often. They only divide when they have to. Okay? If you have a proto-oncogene in your liver that gets turned on always, always on, then that means that you are going to have, even if it's healthy liver cells, those healthy liver cells are going to, you're going to have a buildup of cells. And that buildup of cells is going to create a tumor. Now, how does that become cancer? Well, because you're continually going through this cycle over and over and over and over again, nobody's regulating what that DNA looks like. So each one of these checkpoints, the proto-oncogene just goes, oh wait, I want to put it in purple. The proto-oncogene just goes, oh man, don't worry about it, don't worry about it, just go on, just go on. All right? It doesn't even regulate it. It's not even looking at anything. It's going, just go on, go on, go on, go on. It's fine, it's fine, it's fine. So most likely the cells that are being made are mutated ones. They're not right. There's something wrong with them. But the proto-oncogene's been like, no, you go on ahead. It's fine. All right? So that's one way. Another way is that we have something called a tumor suppressor gene. So I'm going to write that in pink. Tumor suppressor gene. And when the tumor, this is a negative regulator. This tells the cell to stop dividing. Okay? So, for instance, proto-oncogenes are on a lot in your skin cells because your skin cells always need to divide. But in some cells, like your liver is going to go between tumor suppressors and proto-oncogenes. Okay? Depending on if it needs to regulate or not. Okay, so a tumor suppressor is going to prevent, when it's turned on, it's going to tell them to stop. So this is the opposite of the proto-oncogene. Tumor suppressor, when it's on, no cell division. It's the opposite. When it's off, then you have all the cell division that you want. Okay? So if I get to this checkpoint, and let me erase the checkpoints. And if I get to this checkpoint in a normal cell, if I get to this checkpoint, this first one, if I get to this checkpoint and it's not right, then a tumor suppressor gene is going to stop it from dividing. That's its job. It says, no, we're not going any further. Okay? Now, we already talked about how even if a tumor suppressor gene doesn't want to, this messed up proto-oncogene can come over here and say, hey, we're going to keep going, go, go, go. But what if there was a mutation and the tumor suppressor gene was turned off? So the tumor suppressor gene, a tumor suppressor gene was never turned on. So this messed up gene, this is all messed up, and it gets here, and tumor suppressor isn't there to stop it. It's turned off. Then guess what? We can just unregulated cell growth. We can just grow and grow and divide and divide and divide until we end up with cancer. Okay? So again, so a tumor suppressor in real life, in a regular cell, is supposed to stop cell division. A proto-oncogene in a regular cell is supposed to start or continue cell division. All right? The way that cancer sneaks by is, I thought I turned it to green. Cancer is going to stop these from doing their job, which means we're going to create too many cells, too many cells that have not been checked. So they probably have mistakes. So you're going to have too many cells that are bad. Since they have not been checked, they're probably mutated.